Hello everyone, welcome back to our channel. Today the topic of our video is one of the cancer early screening techniques, genetic testing technology. In this video, we will discuss the workflow and application scenarios of genetic testing technology. So let's start today's video. Cancer usually plays hide and seek, popping up like an unwelcome surprise party just when we least expect it. But guess what? Our sneaky enemy often leaves behind clues in the form of specific genetic hiccups. That's why we've got our trusty genetic detectives on the case, using high-tech genetic testing to unmask these troublemaking genes early and give a cancer a run for its money. Common genetic testing methods include genome sequencing, gene chips, and fluorescence in situ hybridization. Think of gene chips as a lively DNA matchmaking party. These chips are like a bustling arena, with thousands or even millions of eager little DNA matchmakers, each holding a card that represents a famous gene, a unique DNA snippet, or even a genetic secret code, SNP. They're all here to make love connections through complementary pairings and wild hybridization reactions, helping us unravel the genetic mysteries of life. After a sample, RNA or DNA, undergoes a hybridization reaction with the DNA probes on the chip, it is possible to determine the expression level of each gene or the SNP status in the sample. Fluorescence in situ hybridization is like a genetic treasure hunt in the microscopic world. Picture this. We send in our team of DNA detectives armed with tiny, colorful flashlights, fluorescently labeled probes, and they're on a mission to find the secret genetic codes hidden within cells or tissues. When they spot their target DNA or RNA sequences, it's like a disco party under the microscope as the probes and genes do a neon dance together, revealing the genetic map we've been searching for. If the probes pair with the target sequences, they will bind together to form a fluorescently labeled probe-target complex. Gene sequencing is the Sherlock Holmes of the genetic world, always on the hunt for clues about our DNA or RNA. It's like reading a thrilling genetic novel, searching every page for typos or plot twists in the base pair storyline. Common sequencing methods include Sanger sequencing, next-generation sequencing, single-molecule sequencing, and others. The differences among these methods lie in the sequencing speed, cost, and applicability, among other factors. Currently, early cancer screening services are primarily conducted through second-generation sequencing. Therefore, in addition to traditional sequencing methods, with the development of sequencing technology, many new approaches are being applied in multi-cancer early detection. For example, detecting circulating tumor cells, CTCs, in the blood, or circulating tumor-free DNA, cfDNA. Early screening aims to detect cancer genome characteristics that differ from the normal background genome. Circulating free DNA, cfDNA, is composed of degraded DNA molecules released from cells into the bloodstream. It is typically present in low concentrations in the blood of healthy individuals. However, when a patient develops cancer, cells release a significant amount of DNA into the bloodstream, leading to a general increase in the levels of cfDNA in the blood. Compared to traditional detection methods, ctDNA, circulating tumor DNA, testing offers several advantages. Convenient sampling. It only requires a blood sample to analyze tumor cell DNA, avoiding the risks associated with procedures like biopsies and surgeries. Mature detection technology. Utilizing next-generation sequencing to test cfDNA, it provides high sensitivity, accuracy, and low false positive rates, capable of detecting mutations as low as 0.1%. Comprehensive detection. Virtually all tumor cells release DNA into the bloodstream, and cfDNA can reflect the overall status of the tumor within the patient's body. Steps for cfDNA sequencing. 1. DNA extraction and detection. 2. Library construction and quality control, including methylome analysis. 3. High-throughput sequencing on the machine. 4. Bioinformatics analysis. Genetic testing plays a crucial role in cancer early screening and can accomplish the following. 1. Cancer risk assessment. 2. Early cancer screening. 3. Personalized treatment customization. 4. Prediction of drug responses. 5. Family risk assessment. 6. Clinical research. In a nutshell, think of genetic testing for cancer as a gene-themed game of spot the difference. We're like genetic detectives with a magnifying glass, scrutinizing your genetic code to find those sneaky mutations that might make cancer think it's invited into the party. Armed with this insider info, we can then craft personalized cancer-finding strategies, ensuring that every patient gets their own unique superhero cape in the battle against the cancer.